Thank you for joining us today. I'm Connie Yu. I'm a curator on the UniProt team. My expertise is in structural biology, and I will be doing most of the talking today. We've also got Emily here with us. She's also a UniProt curator and a proteomics expert on our team. She'll be helping out behind the scene, making sure everything runs smoothly. So, just a little bit about myself. I've been a structural biologist for over 10 years. I had my postdoctoral training at UCL and later worked as a staff scientist at the LMB before joining UniProt. Over the years, I've worked in a range of research areas from protein translation, RNA transcription to cell division. And across all this work, my focus has always been on using structural data to understand biological mechanisms, which is also the focus of our webinar today, how to get biological insights through structure function analysis. So this is an overview of today's webinar. I'll start with an introduction about protein structures and UniProt. Then we will do a walkthrough on how to find structures on UniProt. I will highlight how structural information are captured and annotated in UniProt. Then finally, we will have a brief demonstration on how to assess this data programmatically before going to the conclusion and Q&A. So first thing first, why are we interested in protein structures? So we know proteins as molecular machines. They have a sequence that defines their 3D structure, and that structure defines what they do. Protein structures allow us to know how a protein folds, where it binds substrate, or which residues forms the active site. And that enables us to interpret almost everything about its function, how regulation happens, why a mutation causes disease, and how we can design drugs that target those diseases. So the key here is we want to link structure and function because that allows us to translate raw sequence data into biological understanding. So why UniProt then? UniProt is the world's leading high-quality, comprehensive, and most importantly, freely accessible resource of protein sequence and functional information. What I want to highlight to you today is that we also have a very extensive structural coverage. With over 140,000 experimentally determined structures mapped to our entries, UniProt is one of the world's most complete structure-annotated protein databases. Through collaboration with the AlphaFold database, we also provide over 200 million predicted structures for our protein entries. And recently, this collection has further expanded to include various predicted structures and models from the 3D Beacon network. So the 3D Beacon is a collaboration between providers of macromolecular structural models that include experimentally determined structures from small angle scattering biological data bank, a template-based model from AlphaFuel and Swiss model, conformational ensembles from Protein Ensemble database, and also ab initio models. Of course, AlphaFold is also one of them, and then isoforms.io, model archive, etc. So it's really a very extensive collection of structural data, and that means structural information is always available for any protein entry in the UniProt knowledge base. So how do we look after this huge structural collection? We have a collaborative project between the UniProt and PDB Europe called SIFS, which stands for Structure Integration with Function, Taxonomy, and Sequences. The aim of SIFS is to provide consistent annotation in different structure entries and to support accurate and up-to-date cross-reference information. What that means is when you're looking at an amino acid in a protein sequence in UniProt, you can expect to find the same amino acid in all the corresponding structure. So let's say if you find a lysine in position 90, you will expect to find the same lysine in the position 90 in a structure. And that is crucial for transferring annotations between protein structures and sequences. It allows you to confidently explore information about a particular amino acid across our extensive cross-reference database. Again, using the same example, if you want to find out more about translation or modification of this residue, you can go to our cross-reference database, knowing that you are examining the same amino acid position in each context. Okay, then let's get started with our walkthrough. This is the UniProt homepage. If you have a spare screen or if you have spare space on your screen, you're invited to go to the UniProt website and do this walkthrough with me. We will start with a simple search for protein you can use its common name or gene name, but in this example, I'll be very specific and use the UniProt accession ID. So I'm using Q12834. Clicking search will bring me directly to the entry page. So this shows you the top of the entry page. Under the blue menu bar, you will see in large letters and in bold the UniProt accession Q12834, which is what we were searching for, and the identifier of this protein, which is a human cell division cycle protein known as CDC20. On the top of the page, you'll find some basic information about the protein, include the protein name, gene name, taxonomy, its length, how many amino acids are there. On the left-hand side, you will see different sections with biological information about this protein. These sections are clickable, and when you click them, it will slap you down the page directly to that section. So here we will click and go to the structure section. So if you're doing this on the website, on the browser, you should see the structure viewer on top and the table underneath. I've rearranged them to fit them on my slide. But here we are looking at the structure viewer. Underneath is the table with all the structures and predicted models of this protein. You can click different rows to select different structures or models, and they will be shown on the structure viewer. The experimentally determined structures from PreDB are always at the top of the table, but you can also filter them using the source drop-down menu and choose PDB. You can also do filtering using the method drop-down menu, which allows you to filter based on the methodology used to obtain the structure. For PDB structures, the most common methods are X-ray, which stands for X-ray crystallography, and EM, which stands for cryo-electron microscopy. When you are selecting a structure to study, there are a couple of things to consider. First of all is the method and the resolution. So for X-ray and EM structures, a lower number here suggests a high resolution, and they are more likely to provide atomic details of the protein. Secondly, you should also consider the amino acid positions. I'll explain this in more detail. So in structural biology, we often use an engineered protein sequence to solve the structure, and this is what we call a construct. The construct will contain your protein of interest, colored here in blue, but it often also contains additional sequences, such as an affinity tag to help you with purification. So this is shown in gray, these extra bits. It might also contain some mutations, like this one highlighted in red. When the structure is being solved, we might not be able to resolve every amino acid in the sequence of the construct. So it is possible that some amino acids cannot be observed because of the methodology or resolution. In UniProt, what we map to is the biologically relevant sequence from the construct. That means two things. First, it does not include any additional sequences, the affinity tag that we've talked about, which are not biologically relevant. Second, this is regardless of whether the sequence is observable in the final structure. So this will be the sequence that we map to. It is quite important to check the amino acid position when you're selecting a structure so that you know you will be looking at a structure that covers the sequence you're interested in. So we've looked at a simple structure with just our protein of interest, but a lot of structures contains multiple proteins, and our protein is only part of that huge complex. In these cases, it is important to know its chain ID. In PDB structures, each entity is given a chain ID. So if you want to explore this protein further in the structure, for example, this one that I've highlighted, now I know it is chain R, and then I can trace my protein using the chain ID. There are also cases where no experimental structures is available, but we can still get structure information from AlphaFold and other predictive models. Particularly for AlphaFold, since they are highly accurate predicted structural models, they can be very informative. In UniProt, the AlphaFold model is always provided for the full-length canonical sequence. And you can also see that different regions of the AlphaFold model is color-coded based on the confidence of the prediction, with blue suggesting higher confidence. So now we have found all the structures of CDC20. If you want to dig deeper, you might also want to read the publications associated with these structures. And to do that, we'll go back to the top of our entry page. Underneath the basic information of the protein is a row of tabs. So we've been looking at the entry tab, and now we are going to look at the publications tab. So if you click that, this tab shows you all the publications that are added to this protein entry. There are a lot of them, but on the left-hand side is a selection of categories. Publications can be filtered by categories, which are different types of information provided by the publications. Clicking on the structure will filter out the 8 publications that provide structural information for this protein. So you can scroll to see the list of publication. Each publication is shown with the title and author list. You can click View Abstract to get a sneak peek of what this paper is about. From the source, you can see each publication is mapped to a different structure. You can click on this identifier, which will direct you to the PDB page for more details. You can also click the link to PubMed or EuroPMC that direct you to these databases, or you can click the journal information, which should bring you directly to the article in that journal. Okay, now we have looked at how to find all the structures of a protein. What if I want to do things the other way around and find all proteins in a structure? This is particularly handy these days as the structures are getting bigger and more complex, and we often find more than one protein in a PDB structure. So take this one as an example. This is PDB 8PQL. I want to know about all the proteins in this complex. So what I can do is, again, do a search in UniProt. So going back to our UniProt homepage, previously we have searched with a UniProt accession ID. This time we're going to search with a PDB identifier. And to do this, we are going to use the advanced search function. So you can find the advanced search button here. If you click that, that should bring up a panel. There are drop-down boxes for you to select different fields, and you can just type in PDB and select PDB as the cross-reference. And then in the text box, we will just put in the PDB identifier 8PQL and click search. This will bring us to a results table based on our search for the PDB identifier. This, again, a reminder that this is the same identifier you see across all the PDB web pages, so you can find the structure of interest and search them in UniProt. The results table here may look slightly different in your browser, as we provide customized columns that allows filtering and personalization. For example, here I've added a 3D structure column so that at a glimpse, I can see the number of structures available for each methodology in each protein entry. Now looking at this table, you can see there are actually 8 different proteins identified in the structure. I can see the protein names, gene names, the length of each protein entry, and I also know this is a human complex from the organism column. If I want to find out more about each protein, I just have to click the UniProt accession to check out the individual entry page. So let's have a closer look at one of them. Let's pick Curlin 2. I'm going to use Curlin 2 as an example to show you how we can learn more about the architecture of a multi-protein complex. So now we're in the Curlin 2 entry page. We will go to the interaction section, and you can select that from the left-hand side menu. At the top of the section, you will see a free text comment that has been manually curated by our team using data extracted from literature and the experimental structures. So this comment usually describes the quaternary structure of a protein, its interacting partners, and its involvement in any biologically relevant complexes. A reminder here is that only stable interactions are recorded in this comment. This means it does not include any transient interactions or transient complexes. It also does not include enzyme-substrate interactions or receptor-ligand interactions, as we consider this as intrinsic part of a protein's function, and they're therefore annotated in the function section instead. You will see a schematic display of this complex if the protein is in a biologically relevant complex. So occasionally, structural biology may capture a protein complex that is biologically irrelevant, meaning there's no evidence that this complex or interactions actually exist physiologically. But if you see this schematic, it suggests that a biologically relevant complex has actually been identified by the complex portal, which is another protein database hosted by the EBI that focuses on protein complex curation. And we can have a closer look at this biologically relevant complex by clicking View Interactors in UniProt KB. If you click that, we can see all the components in this complex. This time you will see a result table from a search using the complex portal ID. Here we can see all the protein components. We know this is a biologically relevant complex, which means now you can actually compare the composition here with the structure that we've looked at to see what proteins are missing or which protein is actually additional in the structure maybe. This really helps you to understand what information is available in that complex structure. So to do that comparison, you may want to download this data for further analysis. For our results table, you can find the download button up here, and you can download this results table in different formats using this button. Okay, so that's the walkthrough. Now I'm going to talk about how structured data are captured in a UniProt entry. So since the structure is very informative in multiple areas, this data can be found across a range of sections in a protein entry. And we'll go through these different sections one by one. I'll be using different protein examples as shown down here. To start with, the function section, there is a subsection called features, and you will see a graphical view of the protein sequence here. We have the yellow dot highlighting the active site and the black bar for the binding sites. This is an interactive display, which means when you click an amino acid, it will take you to that amino acid position in the table below. You can again filter the type of sites that you're interested in using the drop-down menu. And throughout the entry page, you will see evidence text that looks like this. The most common one is the publication tag, suggesting the information comes from a publication. There's also a combined source tag. So if you click it, it tells you that this data comes from a structure. The ATP binding site is actually annotated based on the PDB structure 8F09. And if you click the identifier, it will again bring you to the PDB page for more details. And also, these are all residue-level annotations, so we're only using high-resolution structures to capture this information. Okay. Next, we'll go to the subcellular location. This is where we annotate the protein topology and any transmembrane region of the protein. For this example, you can see the transmembrane helixes, they're annotated, and they're also shown as black bars in this schematic. We also often find mutagenesis data from structural biology papers, and we will annotate them in the diseases and variants section. If they're related to a disease variant, that will also be annotated. We have a PTM/processing section. This is where we capture a lot of post-translational modifications that you can see in the structure, including phosphorylation, glycosylation, cross-links such as ubiquitination or disulfide bonds. And the beauty of this is you can also compare with data from other cross-references, for example, PTM, Exchange, and Pry, and it's really handy to have all this data in the same section. Then we have the family and domain section. This is the section where we capture a lot of structural features, including domains, structural repeats, call. If you're looking for those things, this is really the section to look at. And even though we have been talking about structured proteins, I want to give a special mention to the unstructured regions as well. In UniProt, we also annotate disorder regions and regions with compositional bias. Often these regions have no experimental structure data available, and they're usually computationally annotated using a prediction server. So you can see here the evidence tags actually say automatic annotation, as they're annotated using MobiDB in this example. And so I have quite quickly gone through a lot of biological and structural features and show you how they're all found in different sections. So you may ask, is it possible to see all these features on one page and maybe also on the structure? And of course, the answer is yes. To do that, I'd like to introduce the feature viewer. Like the Publications tab, the feature viewer tab can be found at the top of the entry page. So here. This time I'm going to use another protein as an example. This is another cell cycle protein called MAT1. So let's have a closer look at MAT1 using the feature viewer. The feature viewer is a graphical representation of all the sequence annotated features available for this protein. Everything in this viewer is clickable. At the top is the sequence. This is where you can zoom in and out to different regions. There is a series of tracks. Each is a piece of biological data mapped to the protein sequence. The structure-related tracks are down here. As we have seen previously, the structures may cover different ranges of the sequence depending on the experimental method and the construct used to determine the structure. So the sequence coverage of these different structures, they are shown in the PDB 3D structural coverage track. So you can see in this example, there's experimental structure in the C-terminus, but there's actually no experimental structure available for the protein in the N-terminal region. In this case, you can refer to the AlphaFold model for structural information. Like what we have seen in the structure section where the AlphaFold model is color-coded, the AlphaFold track here is also color-coded in the same way, based on, again, the confidence level of the prediction. So you can choose to look at the different regions accordingly. I also want to use this to look at an example of PTM. So I'm picking a threonine quite close to the C-terminus at position 716. Clicking this residue on the PTM track shows that there is literature evidence that this threonine can be phosphorylated. You'll see when this is being chosen, when I click this amino acid, there's a yellow highlight of this selected amino acid across all tracks, in case you want to go through the other tracks as well. And this amino acid is also highlighted in the structure viewer, which in the browser, you should find it right below the feature viewer. So this is, again, the structure viewer with a table, very similar to what we have seen in the structure section. The feature viewer and the structure viewer, they're linked. So when you click an amino acid on the feature viewer, that amino acid will also be highlighted in the structure viewer. But make sure you have picked the right structure. Some structures may not contain the residue of interest. For example, I picked 716 and it's not included in this highlighted structure. So instead, you'll actually see an error message on screen saying it's out of range. But if I select a different structure that contains threonine 716, I can now see that it is being highlighted in the structure viewer. In this example, you actually see two residues are being highlighted, as MAT1 is found in both chain A and chain B, which means MAP1 actually exists as a homodimer in this structure. So this is really another reminder how useful it is to look at the chain and position information when you're selecting a structure to study. Okay, so all these structure annotations, they are computationally accessible through programmatic interfaces to support high throughput analysis and machine learning pipelines. We are going to go through the same examples we have covered, but instead of using the UniProt webpage, we will do the same task using the UniProt API. There's also an advanced example that we'll not cover today, but I'll leave it to you. So now if you put this link in the browser or use this QR code, that should bring you to the Collab notebook. And if Emily can help to maybe paste that link in the chat as well. So you need a Google account to access the Collab notebook, but I'll show you how we're doing this step by step. So it should be fine. Let's have the browser up. Okay. So this is our Collab notebook. Thanks, Emily. Here are our API examples. As I said, I'm going to leave the last examples to you. There's an introduction in case you come across this page without attending the webinar, but I'll go straight to the API examples. So here's the first one. Again, we are trying to find all the experimentally determined structure of a protein. Suppose we're still very interested in CDC20 and want to know what experimentally determined structures have been reported. So we'll use the UniProt API to find all the PDB entries mapped to this entry. So this is a Python script. At the start, we import the packages that we need to use, in this case, a request. We'll specify the UniProt accession, again, Q12834. We'll write the script to query that UniProt accession at the UniProt API and retrieve the response in JSON format. There are also other formats available. And getting that result, we will extract the PDB identifiers from the PDB cross-reference. So we specify we are interested in the PDB, and then we'll print out the outputs. So if I run this now, it should tell me that these are the PDB entries mapped to this accession. So I've really written the script in this way so that you can change the accession to any protein of interest. And now you can see the output. There is quite a few structures here, and I want to know more about them. So what I want my script to do next is to list out the method that is used to determine the structure, the resolution, the chain ID and positions that we've been talking about. Again, to do that, first we import some packages in Python. We specify the accession again, do the same query. And this time, instead of just extracting the PDB ID, we are extracting the whole entry. And from the entry, we will get information including the PDB ID, method, resolution, and chain. I've put in not available at the start so that in case any information is missing, it will start with not available. But as soon as we extract the data from the API, we'll fill in those blanks. And then, yeah, this is filling in those blanks. And for each column in the data table, we'll put in a header so that the data table will look nice. And then it's really just creating a data table using Panda and display that table. I also included an error message because, of course, it's possible that you have a UniProt accession with no experimental structure. In that case, there won't be any PDB entries. So it's good to have an error message for those cases. And if I click Run now, I get a very neat data table, very similar to the data table we've looked at in our structure section, with all the information we need. So this is how you go through, you find all the structures of a protein. Again, we're doing the same task the other way around, listing all the proteins in the PDB structure. And we will again look at the Q-lin ring E3 ligase that we have looked at using the PDB identifier 8pQL. This time we specify the PDB code. We do another query on UniProt API, a slightly different one, so that we specify the PDB ID, extract the data, and this time we will be extracting the UniProt primary accession as our output. And now you get that you have this UniProt entries mapped to the PDB HPQL, again, the 8 protein components that we've looked at. Again, let's make a data table with information of these protein entries. It's very similar to the script we have looked at. We import the package we need. The aim here is to get the UniProt recommended protein name and organism of each entry. So we specify the PDB ID, query the API, and this time from the PDB, from the UniProt entry, from the UniProt entry, we will extract the accession and also the organism. And again, tidy up the data table, display the data table, keep an error message in case we don't find any protein in this structure. Let's say if you have an RNA structure, there might not be any protein, so it's good to have this error message. And again, you have a data table with all the information you need. Very, very similar to what we have done on the web page. And the whole point of using the API is that, let's say you have 10 or even 100 of UniProt accession or PDB entries that you want to analyze. You can easily modify this script to do this automatically, and this allows you to easily scale up your structure function analysis no matter how many entries you need to look at. So that's the example. As I said, I will leave the third example to you. This is again a scenario where you don't find experimentally determined structures, and then you can try to look at homologs. Okay, with that, I'll go back to the slides. That was a very brief demonstration on how you can programmatically get structure information from UniProt. This is really to get you started. If you need more detailed explanation on programmatic access, we have a UniProt webinar lined up in March. It is an advanced webinar that will introduce the various API we have in UniProt. We have more than one, and they all do different things. You can use them to retrieve protein sequence and other metadata. If you're looking for an integrated use of various resources from the EBI, including UniProt, Interpro, PDBE, AlphaFoldDB, there's another training workshop in June, and more information about this workshop should be available in March. Okay, with that, let me conclude with a summary about the things that we've talked about. So we've talked about how structure information is captured in UniProt, how structures in UniProt can provide residue-level annotations. We talk about the consistent numbering and mapping between UniProt and PDB, which allows you to confidently explore interactions with ligands and active sites, PTM, mutations. We have walked through how to look at architecture of multi-protein complex, and we have also looked at how different topological arrangement are captured in UniProt in all these different sections. We've also used the feature viewer. We've learned how to check proteins, 3D structure coverage, and how you can use AlphaFold predictions and other structural models to assist structural analysis. And hopefully you will find all these resources useful. And if you benefit from the use of UniProt, I'd like to encourage you to help others benefit as well. You can simply do that by providing the UniProt identifier with your data and in your publication. The UniProt identifiers will allow readers and us, the curators, to identify the sequence used in your study. And this will help us annotate your work in UniProt, which in turn will help others discover your research. So to quote my colleague Suchu from the Complex portal, don't just publish, be found, help us make data more accessible and in turn support new research. With that, I'd like to acknowledge the huge team of curators and developers at UniProt who work tirelessly behind the scene to keep this valuable resource running. And with that, I will pass it back to AJ.
Hello, Connie. Thank you very much for this wonderful webinar. And thanks everyone for joining in today and sending your questions. So we have had loads of questions already, Connie. Some of them
Yep.
have already been answered by Emily. But yeah, before we go on to the Q&A, just to let you know about this slide. So there are other webinars coming up from UniProt very soon. And there's also a link to UniProt webinar series, which runs every year. So you can follow there what webinars are coming up. And it keeps on updating as we have new webinars. All right. So with that, we can go into the Q&A session. So the
Uh-huh.
very first question that I can read is how to deposit a new protein structure in UniProt.
Okay, so to deposit a structure, you don't deposit with UniProt, but you can deposit it to the PDB database. Uh, like I said, we have this collaboration with PDB, so any new structures coming to PDB will eventually be passed onto UniProt and we'll map them accordingly. So you don't have to deposit a structure to us. We'll get them directly from PDB.
Very good. The next one is what if a protein has multiple structures or conformations owing to their activation and inactivation states? How does UniProt account for those variations in the database?
So any structures mapped to that protein will be shown in the structure section, as we have shown previously. It is this the different states is not shown in the table, but if the structures has been curated by curator, this will normally be captured in the comment as well. So that is the free text comment I've shown you in the interaction tab. So that's how it is captured.
Okay,
Mm.
thank you. Is there an aggregated view for complexes?
Uh, is there an aggregated view for complexes? Emily, any thoughts?
I'm sorry, I was really engrossed in a question there. Sorry, Connie, can you repeat that?
The question is there any aggregated view for complexes?
You will probably know better than me. I think you've shown everything we've got on the UniProt
Yeah.
website.
I'm not
Yeah.
sure what I've shown is what they're thinking about aggregated view, but that is the view that we have at the moment. That's how we display the complex. Um, yes.
All right. So the next one is what interesting functions does UniProt have for identifying properties related to the protein IDR region, such as electrophysiology?
So like I said, these IDR regions, the issue is really there's not much experimental data available for them. So in most entries, I think what you see is automatic annotation we use MobiDB quite heavily for IDR predictions. Um, I'm not aware we have electrophysiology. I can double check that. But again, if there's any interesting functions that was reported in literature, this will also be captured by curators during curation. Mhm.
Great. Thank you. The next one is about multiple IDs. How can you have multiple UniProt IDs for one PDB ID? I understand the other way around.
So each UniProt ID is for one protein sequence, while each PDB ID is for each experimental structure. And very often in an experimental structure, you actually see more than one protein. So, for example, in the protein complex, you might have three, four, five, or even in ribosome, you have 80 different components in the structure. So that's why you will see multiple UniProt ID in that case.
Okay, great.
As a
Um,
follow-up question.
yeah, I think the next one is follow-up or probably more clarification of the previous question. Sorry, my question is whether UniProt can detect parameters related to the IDR region of proteins with IDR, such as
Hmm.
charge statistics. This would
Hmm.
be very useful as it would provide a direct indication of the protein's likelihood of binding to nucleic acids.
Uh, at the moment, we don't. I am very aware that there are servers out there that can do more analysis on IDL. It's probably something we can look at in the future.
Okay, thank you. One last question, which is in here, is there any intention to include molecular dynamics in UniProt?
Not yet, but I'll make a note that there is interest in this region, this area.
I've made a note.
Yeah,
Very good.
that's why it's good to have webinars.
Yeah.
Very good.
Okay.
So we still have a few minutes left. So if anyone else has any question, feel free to type them in. We are still here.
Um, I just try to elaborate a little bit more on the aggregated view of complex. So if you're thinking about looking at the structure, then like we've said, the structure view is what we have at the moment. I've shown that there is also the complex portal cross-reference. That's also a very handy tool if you want to know more about the complex. So that's something you can go to for more information as well. Recently, there's also an update from the PDBE web pages, and I know they also have a complex page. So if you are really very specifically interested in the complex, those are the resources that you can go to.
And as this is the very first webinar from UniProt for this year, I was just wondering, is there any update or something to highlight for, watch this space thing for UniProt?
So we've got our new release at the end of this month. It's currently planned for the final week of January. And that's our new data release that's coming. We're also going to make it very obvious to people that our reference proteins are changing. Most of our changes happened in the last release, and there's some coming again later in the year. But you'll see quite a bit of literature on the UniProt website regarding that. Please take a read, especially if you're into bacteria and virus work. We've got a tools webinar coming up later this month with Antonia. Just going to check the date on that for you guys. I would advise that you attend the Python webinar if you're into programmatic access, because all this data and more is accessible programmatically. And we're going to walk through examples that you could do on the website and then move them into programmatic access in a lot more detail and give you some examples of how to use that data to then do graphical analysis and various things there as well. So that webinar is on the 5th of March. And that's the programmatic webinar, and our tools webinar that's open for registration is on the 22nd of January. So that will cover BLAST and ALIGN and our protein analysis tools that we run from UniProt as well.
Very good. So yeah, that has prompted many questions that came in. So can you comment on UniProt's protein sequence and structure alignment?
Not sure which alignment function we're talking about, Emily.
If you want to align sequences to one another, you use the align tool that you find in your top toolbar on the UniProt website. If you're aligning a sequence to a structure, that's probably more for you to comment on, Connie. But you can query a UniProt database using BLAST and align for that as well, and then look at structural data.
Yes. So, okay. I hope I'm getting this correct. Yes, aligning the protein sequence, like Emily said, is more easily done. If you want to do a structure alignment, that's a bit beyond UniProt, but what you can do is actually there is something you can do on UniProt, which reminds me, because we also have a link to FOSseek in the structure section. I don't know. There we are. So a very good tool to do the structural alignment or structural search is using this FOSseek tool, especially if you want to find similar structures. And of course, another way to do this is actually download the structures and do it using more specific tools like Pymo or Chimera to do the structural alignment, if I understand the question correctly.
Yeah, thank you.
Okay.
Next one is, if I understood correctly, I cannot find protein-protein interaction or putative partners for my protein of interest on UniProt.
You should be able to find protein-protein interactions for your protein of interest in UniProt, unless we haven't come across that reference or the literature reference that prove that protein-protein interactions. But again, I always like to emphasize that in UniProt we captured stable interactions that has evidence that they exist physiologically. So if you're talking about a more transient interactions, we won't have that annotated in entry.
Thank you. UniProt provides structures that are not provided on PDB. Also, can we consider protein structures provided by AlphaFold DB for further analysis like molecular docking?
So to clarify, we provide structures from PDB that are experimentally determined. There are other structures provided that they are more predicted model or there are predictions or models. And of course, you can also use them for analysis. Like the PDB structures, you can just download them and you can use them for any other further analysis, including molecular docking.
In UniProt, is it possible to see the associated compounds to certain proteins? For example, inhibitors of ion channels.
Again, that depends on whether the compounds we're talking about are biologically relevant. I'm still trying to think from the structural context. So in the structures, very often you will see multiple compounds bound to a structure. Some of them may just be for the stabilization of the protein during structural biology. It might not actually be biologically relevant, so they might not be captured in our annotations. But if they're biologically relevant, then again, you would see we'll annotate them in the ligand. There's a ligand binding for this protein.
Next one is a bit related, I think. Does UniProt provide interaction of the target searched protein with other proteins? And if so, then how does one gets to know which protein shows good interaction?
Again, trying to emphasize that we do capture interactions that are physiologically relevant and stable interactions. It's almost impossible to say which one is a stronger binder in that context. So all I can say is we'll capture the interactions. And if you really want to explore the interactions, the affinity in more details, maybe you have to refer to the reference. Unless it's something very specific, then we might have annotated that.